All right, in this video, I'm going to be talking about something called splicing. All right, now splicing is something where you alter the mRNA before it reaches the ribosome. So the mRNA has been created during transcription. All right, so let's start with transcription just to remind you. So you have DNA, and so we all know that DNA is double-stranded, represented very simply nucleic acid structure. Right now, only one of these strands, though, something called RNA polymerase. You watch that. Copies this either two strands. All right. We'll talk about the direct later. This is going to be copied in RNA. A copy has been made of RNA polymerase attaching to the DNA and making this RNA here. Well, this RNA normally, like in a bacteria, you would send this RNA, or I should say the ribosome will be sent to the RNA. At some point, you're going to have this RNA meet up with a ribosome. So a ribosome here, it's actually got two parts. Think of it as a large part and a small part, and it attaches to the RNA. And when these two pieces attach here, all right, they are going to be reading that RNA and producing an amino acid sequence. All right, so all these little pieces are amino acids, and they're all attached. All right, not too different from RNA. It's much larger actually than RNA. These are amino acids, and basically you read the instructions along here. So this ribosome will attach and read the instructions along the RNA, and it assembles this, what we call a polypeptide. It's a bunch of amino acids bonded together. All right, and then of course this polypeptide, this set of amino acids that are produced from this, it folds up. It kind of curls up on itself. It's a chemical process called folding, and you get a functional protein from this. So you start with DNA up here. So here's your DNA. It's hard to write on these, but DNA. Then you make an RNA. All right, and then you go to the ribosome, or the ribosome goes to the RNA, depending on the way you look at it. If it's a prokaryote, generally you think of the ribosome going to the RNA. If you think in terms of eukaryotes, you have the RNA leave the nucleus and go find the ribosome. So this would be RNA here, but this is this piece. These are amino acids. This is an amino acid sequence. All right, and we kind of call that a polypeptide. And when it folds up, we call it a protein. Of course, the protein does all the work inside the cell. All the chemical work is primarily done by proteins. So these are the instructions. These are a copy of the instructions. The assembly mechanism, like a factory, is that reads the instructions and puts them together. That's the ribosome, and then the protein itself is what does the chemical work in your body. So amino acid sequence, I know that's hard to read, but that's the key. This is the standard procedure, but there are a few other steps that we need to add for eukaryotes. So I'm going to go over here and we're going to make a new picture here. And what we're going to do is we're going to say, yeah, here we start up here with DNA. There we go, DNA. And yes, we do make that single-stranded copy in the form of an RNA, but we're going to call this it's hard to get letters right here. All right, we're going to call this an immature RNA. So let's see if I can take my time here and say immature RNA. Well, eventually we are going to get down here, and it's going to be read by the ribosome. The ribosome is going to read it and make the polypeptide, which is going to end up being your protein. But in between here, in eukaryotes, all right, in eukaryotes, there's going to be another step. And that step is, there's something called the spliceosome. It's a set of proteins. So let me write this. It's a set of proteins and some RNAs that kind of get together. Think of like, it's a bunch of proteins that kind of attach to one another, and they also have some RNAs inside of them. So I always think of it as like if you're taking a couple of pieces of pre-chewed chewing gum, stick them together and add some cat hair, and you've kind of got this mess. So a bunch of proteins. All right, I know that's a very graphic thing. And then the cat hair are some other RNAs that are made, but they're not the type of RNA that you make from your standard gene. These are special RNAs that are part of this thing called the spliceosome. Not too different from a ribosome, to tell you the truth. A simpler structure, but essentially this spliceosome can attach, all right, to certain areas in these immature RNAs. We're going to call the one down here that gets to actually to the ribosome, or the ribosome attaches to, actually I should say it does. It would be the one that gets to the, it would move in a eukaryote, it would move out of the nucleus and into the cytoplasm. So this RNA down here, this would be called a mature RNA. So really what we're saying here, the spliceosome, let me see if I can grab this, mature RNA. All right. How do you go from immature to mature? Well, that's what the spliceosome does. It splices things out. It actually takes pieces of this RNA, all right, and it attaches here. It creates little loops, all right, so it kind of loops the DNA, let's say here, loops it around, and this is the spliceosome here, attaches, and it will cut a piece out and make these two pieces. So this piece here, all right, and this piece here get attached, and this piece gets cut off. So this little loop here goes its direction, all right, and is destroyed. And then these two pieces here and then this little piece here, they're attached together. And so you cut out these pieces. So there might be several of these pieces that are cut out, all right, maybe the majority of the sequence. In fact, most of the time, majority of the sequence is cut out. So these regions get chopped out. And so let's label these. Let's say A, B, and C. Well, you're going to take A, B, and C, chop out the two X's. We'll just go X1 and X2. Take out the X1 and X2 in this process. And of course, what you get is a piece that's got A, B, and C in it. And this is your mature RNA. This is the one that goes down here and associates with a ribosome. So we spliced out these two pieces. They are cut out, all right, and the pieces left over are attached to one another in this process I drew over here. So in splicing, all right, we call this the immature RNA. We go to the spliceosome and we turn it into a mature RNA. Now realize this occurs in eukaryotes, organisms like ourselves. This does not occur, at least it doesn't commonly occur. There actually is a case where there are some proteins that can do something like this in bacteria, in prokaryotes, but it's not the same thing as here. So this is in eukaryotes, so keep that in mind. This is what we do. This would also be what flies do. We have to account for these because we will often be working with what we call genomic DNA, and genomic DNA would be this here, and it doesn't have all the splicing sites, but we're looking for the piece of DNA that codes for this protein. Well, we might know the sequence of this protein, and we may know the genetic code, but we have this problem of these gaps in here, and we have to worry about getting these pieces out. So there are some names we need to know. The pieces that you keep are called exons, all right? Exons. So this is exon A. This is exon B, exon C. All right, the pieces you cut out are the introns. So intron X1 and intron X2. And you're going to be splicing. Splicing cuts the introns out and reattaches the exons together. Now, one sequence, one gene, if we consider this a gene up here, watch my previous video on the definition of a gene. If this is a gene, this region in here, then this gene produces this protein. But does it produce just this protein? Realize there are things going on in the cell that can alter the splicing. So sometimes you cut out X1 but not X2. So that's going to create a different sequence and therefore create a different protein. Sometimes you cut out X2 but leave X1. That again would make a different protein. All right, and sometimes maybe you leave them in or you cut both out. So there are two more proteins. So if you have two introns in your gene, you potentially have 4 proteins that you can make there. You have one where you leave everything in, and then you might cut one out, then you might cut the other one out, all right, or you might cut both. All right, so you potentially, depending on how the splicing goes, you might be able from one gene to create 4 different proteins. And so this throws a monkey wrench in your analysis a lot of times. And you'll have variants, what they'll call variants, and they're not alleles. Maybe there are people who have mutations so that they never cut out one, or they never cut out either of them, or they always cut out both. And that's a different type of mutation than the type we normally think of. But these variants, there may be people who can make 4 proteins from one gene. There may be other people who can only make 1 or 2. And so that each of these may have different functions, and some of these might function better in certain circumstances and worse than the others. Sometimes this might cause a disease, depending on what's going on here and the splicing of your gene. It's an interesting concept, and we need to understand how to account for it when we sequence genomic DNA and we think there's a gene somewhere. All right, we have to account for splicing. So that's what we're kind of looking at in one of our exercises.